So let's focus in on the Krebs cycle. So now let's look at the details of the Krebs cycle. Again, it gets its name cycle because it's a metabolic pathway where we regenerate our starting compound. So notice here, oxaloacetate is a 4-carbon starting compound, and it is regenerated. This blue line that is going around is basically summarizing a series of enzymatic steps. If all we did was regenerate our starting compound, it would kind of be pointless. I mean, why just keep regenerating what we started it with without getting any benefit from it? So our benefit that we get are shown in yellow. We benefit by getting NADH molecules, FADH2, as well as ATP. There is a preparatory step, which is shown up here. For our pyruvate to be able to enter the Krebs cycle, it needs to gain what's called a coenzyme A. So notice the 3-carbon pyruvate molecule bonds with coenzyme A, loses a carbon in carbon dioxide, forming acetyl-CoA. And to assist that process, our NAD plus molecule is reduced to NADH. The NADH will then go to the electron transport system. Remember, from glycolysis we had 2 pyruvate, so this is repeated twice for every glucose molecule. So now we have an acetyl-CoA molecule made per pyruvate. Let's follow what happens to one. One acetyl-CoA molecule is combined with one oxaloacetate molecule. Oxaloacetate is 4 carbons, acetyl-CoA is 2. That makes a total of a 6-carbon compound called citrate with the removal of coenzyme A. Coenzyme A will be recycled and reused in the preparatory process. The citrate molecule, which is 6 carbon, is going to lose a carbon in carbon dioxide and reduce NAD plus to NADH. NADH will go to the electron transport system. It again loses another carbon in carbon dioxide and performs another reduction reaction. Carbon dioxide is given off as waste. That's what we breathe out. The NADH will go to the electron transport system. In the final stages of the Krebs cycle, to regenerate our 4-carbon oxaloacetate, we will make ATP, which can go and do cellular work, another electron carrier FADH2, and another NADH. All of our NADH molecules that are produced, as well as our FADH2, will go onto the electron transport system. The carbon dioxides are waste, metabolic waste that we eventually breathe out. The ATP is used for cellular work. So to summarize, 2 pyruvate per glucose molecule come in to a preparatory state. The coenzyme A is added and a CO2 is lost. This would be per pyruvate. So we end up per pyruvate producing a CO2 and combining a coenzyme A, forming acetyl-CoA. Acetyl-CoA is technically the molecule that enters the Krebs cycle, and as it goes through the Krebs cycle, more carbon dioxide is produced, ATP is produced, and the electron carriers NADH and FADH2. So if we stopped here, we would have gotten some energy out of our glucose, but it wouldn't be a lot. We really didn't get a lot of ATP out of this process. So where do we get the rest? Is electron transport. So the electron transport process, also known as electron transfer phosphorylation, also known as oxidative phosphorylation, is going to happen inside of the mitochondrial membranes. So the NADH and FADH2 molecules that we produced in the previous steps, they're going to donate their electrons to a series of protein-bound molecules that are bound within the membrane. And as they're passed through these molecules, that's called an electron flow, they lose energy, and that energy is going to be used to move hydrogen ions, very similar to what we saw in photosynthesis. These hydrogen ion molecules will build up in the intermembrane space. That creates potential energy that will then be used to drive an enzyme called ATP synthase. The hydrogen ion gradient drives that enzyme to use energy to join an inorganic phosphate to ADP, forming ATP. The hydrogen ions will then combine with oxygen as our final electron acceptor, forming what we know as metabolic water. So looking at the three processes, glycolysis, Krebs cycle, and electron transfer phosphorylation, collectively they work to produce a large amount of energy in our cells. In summary of the electron transport phosphorylation, we're taking the energy from NADH and FADH2 and using it to produce ATP. Oxygen is our final electron acceptor with the production of water. Here's a huge, kind of confusing picture if you just look at it right away, but if you break it down into its pieces, we can see this is a summary picture of the entire process. Glycolysis takes our 6-carbon glucose, puts it into a usable form by the mitochondria called pyruvate. Each pyruvate is 3 carbons, so we have 2. We gain, through this process, a net of 2 ATP and 2 NADH molecules. The 2 ATP can directly perform cellular work. The pyruvate as well as the NADH enter the mitochondria. The pyruvate are prepared to go through the Krebs cycle by losing a carbon dioxide and gaining a coenzyme. So since we're putting 2 pyruvate in and getting 2 acetyl-CoA out, we lose 2 carbon dioxide. The acetyl-CoA then go through the Krebs cycle, regenerating as they go oxaloacetate, breaking the rest of the carbons down, so we end up with 4 carbon dioxide as our waste product. We do get some energy out of the Krebs cycle, 2 ATP. We also get more electron carriers. The 2 NADH that we got from glycolysis, 2 NADH that we got from taking the acetyl-CoA, the pyruvate, and producing acetyl-CoA, and then 6 NADH as well as 2 FADH2 from the Krebs cycle. All of these electron carriers will pass their electrons through the mitochondrial membrane, generating a hydrogen ion in that inner membrane space. A hydrogen ion gradient, a concentration gradient, is generated. The electrons will be received by oxygen as well as hydrogens forming water. And all of these hydrogen that we've pumped into this space have a lot of potential energy that will be utilized by the ATP synthase molecule to, in the end, produce 32 ATP. So notice in summary, through glycolysis, Krebs, and electron transport, we've completely broken our 6 carbons off of our glucose. Here's 4 and 2, that makes 6. And we've generated 4 plus 2 plus 32 ATP. So we have gotten quite a bit of energy out of our 1 glucose molecule. Although yes, energy is lost as we go through each step. Energy is lost as heat. It still is relatively efficient for us to be able to obviously function and our cells be able to stay alive. In summary, we don't have to just put glucose through this process. All of the foods that we eat, the fats, the carbohydrates, and the proteins can all be funneled through aerobic cellular respiration. And this slide, in essence, summarizes where they enter. We know that our simple sugar glucose enters directly through glycolysis. We know that fats are made up of 3 fatty acids, typically made up of 3 fatty acids and glycerol. So these are broken down into molecules that will, from our fatty acids, they're broken into 2 carbon complexes. So they will enter at the Krebs cycle as acetyl-CoA. The glycerol becomes an intermediate for glycolysis, so it enters at glycolysis. And our amino acids from our proteins are broken down into pyruvate molecules, acetyl-CoA molecules, or intermediates of the Krebs cycle. So in essence, our food that we eat, our lipids, our sugars, and our proteins can all become energy for us through glycolysis, Krebs, and electron transport. This concludes the presentation on Chapter 7. I hope that this has been of benefit to you, and I look forward to you attending the presentations for the final 2 weeks. Thank you.